So this is the Stress and Sleep Symposium. We wanted to put together a symposium that looked at the interaction of stress and sleep across human and animal studies. As you've heard about earlier today, sleep is increasingly something that's studied with digital phenotyping. And that's actually something you can do, especially the movement-related measures that we learned about this morning, that you can do across species. So we've put together a panel of people who represent that. And after I tell you a little bit about the data from my lab, we'll have the panel who's here. I'm going to introduce them briefly so that I'm not jumping back up here every time. We have Christine So, who has come from Washington State University, where she's an assistant professor of psychology. And Christine studies disrupted sleep in clinical studies and how disrupted sleep may exacerbate or precipitate psychopathology. She uses a number of different assessment modalities, including ambulatory technologies that she'll tell us about today. Then we'll have Nisa Ibrahim, who is from the University of Washington out in St. Louis, and she's a postdoctoral fellow there. She's going to take us into the animal model world and talk about chronic pain. She studies how chronic pain impacts sleep and opioid-seeking behavior in rodents. Then Rahimi, who is an instructor at McLean and Harvard Medical School, is going to tell us, bring us back again to the humans. Habib collaborates with many of us, and he has deep expertise in these methods. So he'll take you deep into the technology, and he's in general very interested in extracting features from actigraphy and other kinds of digital phenotyping approaches to predict psychopathology in humans, particularly with regards to depression, which is what he'll focus on today. And then we'll have Bill Carlezon as our moderator. Bill, who's a professor of psychiatry at Harvard Medical School and McLean, and the chief of basic neuroscience research. So his lab really focuses on developing animal models of tractable behaviors that are translatable to humans, including in the realm of sleep and stress. So he collaborates with a lot of us human researchers, so he's great to pull this all together for us and move us into the Q&A. I'm Isabelle Rosso. I'm at McLean at Harvard. I know most of the people in this room. So I'm going to get started and tell you a little bit about the data that we're finding in relation, looking at how stress peptides may be interacting and predicting sleep fragmentation in our PTSD sample. And I'll be talking about really our actigraphy-based measures. And as many of you know, these data are part of a larger P50 project that NIMH has funded the past several years that was led by Bill Carlezon and Carrie Ressler, really, and developed to translate measures of arousal or vigilance states and so forth across models of stress-related pathology. So, sleep disruption in PTSD is extremely common and it's extremely debilitating. It's a super prevalent symptom in the acute aftermath of trauma, just as it is in people with chronic PTSD. And depending on the study that you look at, you'll find that prevalence estimates in PTSD are 50 to 90% of patients who report sleep disruption. This looks like insomnia most of the time, as well as trauma-related nightmares. These are also among the most distressing PTSD symptoms, and a leading reason that people actually seek treatment for PTSD is, I'm not sleeping well, much more so than actually the other symptoms. Another reason they're really important is that sleep symptoms are predictive. They're predictive of the development of other symptoms after trauma exposure, and they're predictive of worsening of symptoms in chronically ill patients over time. So we know that they're a perpetuating and a prognostic factor in the illness course. We know also, though, that they're a modifiable factor. So we're increasingly developing targeted treatments to go after sleep in this illness, and I think people will agree that we need more of them, because the allure, of course, is that maybe you help the other symptoms too. So PTSD-related sleep disruption across the board suggests excessive arousal as one of the potential mechanisms. And if you look at all the different literatures with the different approaches that people have used to study sleep in PTSD, they all sort of arrive at this conclusion in their discussion sections. We have the clinical self-report papers, and of course there we have the symptoms of insomnia and nightmares that are embedded into our clinical criteria. And we ask our patients to report on how often they're having these symptoms and how much they get in the way of their life. So they will describe trouble falling and staying asleep, their sleep is super restless, they're moving around a lot, they wake up at night, they're sweating, and oftentimes they're not just going back to sleep, they're getting up, there's hypervigilance, they're getting up and patrolling the perimeter of their house, checking their locks, that sort of thing. So it suggests really this excess arousal, right, by nature. We know that self-report can only get us so far. It's really hard to report on how you've been sleeping in terms of a nuanced way. PSG, polysomnography, has been used in PTSD too. It is considered a gold standard metric to characterize sleep dysfunction. It has a lot of depth. You get a lot of information from it. And the more consistent findings in this literature are that you can find patients show delayed onset of sleep, they have more nocturnal awakenings, and overall you get this rich information about sleep stages with PSG, right? And overall the pattern is that patients spend less time in those light sleep stages, like sleep stage 1, and they spend more time in those light sleep stages and a lot less time in the deep sleep stages. The REM sleep findings, the more consistent ones seem to be of increased REM density, more rapid eye movements during REM, which again people have taken as suggesting excessive arousal, overall pattern of excessive arousal. So the disadvantage, of course, of PSG is that it's in the lab usually, and you're hooked up to all these devices, and most people don't sleep quite the way they do at home when they're hooked up like that. Some people sleep worse. And actually, if you look at some of the PTSD studies, some people with PTSD sleep a little better because they feel safer in the lab. So it's not necessarily as representative. And so in that context, actigraphy has emerged as a nice alternative tool for assessing sleep more naturalistically in PTSD. And by and large, this literature also shows high nocturnal activity movement levels, more frequent awakenings, and less continuity of sleep, more fragmented sleep. With actigraphy, you get movement, you don't get those deep information about REM and non-REM sleep stages, but it's unobtrusive, right? People can use it in their everyday life, and you can give it to people and they can use it for days and weeks and weeks and weeks unobtrusively. So you get data longitudinally if you need it, and you get data about variability, which is important in conditions that have a lot of symptom variability. So if we think that arousal is implicated in sleep and PTSD, what might underlie that? In the Spirit P50, what we've been focusing on are two peptide pathways, hormone pathways, the cortisol and the PACAP pathway. They've generally been implicated in arousal physiology, and they specifically have been implicated in PTSD. So why might they be involved in sleep disruption in PTSD? Cortisol, I'll start with that one, everybody knows about that one, is implicated in sleep in part because it shows a nice circadian rhythm. It's typically highest when you wake up in the morning, it declines during the day, and it's lowest when you go to sleep. So it's tied to the sleep-wake rhythm pretty nicely. It's very sensitive to stress, of course. We all know that the HPA axis engages when we're stressed, starting at the hypothalamus. This is where I was going to take my pointer. Yes. Ooh. Yeah, so starting at the hypothalamus, you have CRF cells that release CRF, and that trigger the anterior pituitary to release ACTH, which gets to the adrenal gland and releases cortisol into your body, and cortisol can do all sorts of things on all sorts of organs in your body to help you respond to stress. Cortisol can also feed back into the brain nicely and dampen down that stress response adaptively, acutely. If you have that happen over and over again, you can get excessive cortisol. And interestingly, though, if you have it happen chronically, you see oftentimes a blunting of this with low cortisol and impaired negative feedback and so forth. And in PTSD, people have, again, literature reviews will say that that is the most common pattern, that we see high CRF and low cortisol. This literature is actually pretty messy. And you can find studies that find high cortisol predicting worse symptoms, and others that find low cortisol predicting worse symptoms, and others that find no difference in cortisol between patients and controls. But there's something there overall, right, in terms of stress. Now, PACAP is pituitary adenylate cyclase activating peptide, and it is also released in the blood, and you can measure it in the blood. It has been implicated in sleep. Predominantly in rodent studies, if you infuse PACAP into the brain, it shifts sleep patterns, and we know that it acts in parts of the hypothalamus that are involved in circadian rhythms, and it can shift circadian rhythms when infused there. We also know that it increases with various types of stress in animals, whether that's restraint stress or acoustic startle stress and so forth. And in PTSD, the person who's done the most of that, oh, he's not here anymore, he left. But Carey, he wasn't supposed to. Carey has done the most research in this and has shown that PACAP correlates with arousal phenotypes in PTSD, including hyperarousal symptoms and acoustic startle. So independently, there's pretty strong literature. What's going to be a little newer about what we've done is we're trying to look at the interaction. And why would we suspect an interaction in relation to sleep? Again, the animal literature has shown us that PACAP can modulate the HPA axis, and a lot of that occurs at the hypothalamic level. It can increase CRF release and then lead to increased cortisol, so there's synergies there potentially. So could that be picked up with actigraphy and blood measures in PTSD? Here's our study. You're going to see data from 135 adults who had trauma exposure in PTSD. 91 of them are women. And we have first thing during their study visit after informed consent is to do a fasting morning blood draw for CORT and PACAP, and to give them one of those gene active wristbands that you heard about this morning, which they wear continuously for 14 days, day and night. And we're interested in the movement that we get from that. Habib is going to talk a lot about the data processing. We're really lucky to have him and Justin do this for us. I'll just say, since his talk is going to be about this, that we take raw activity levels and they get transformed to relative activity levels for each participant for each 24-hour period. And we find the sleep episode in each 24-hour period, because that's how humans sleep, as one consolidated sleep episode. And we want to look at bouts of sleep in the data I'm going to show you. So are there periods where the person's moving and presumably waking up, like here? It goes from blue, low activity, to orange. So this might be a wake bout that we count. We can count them and we can measure their duration. And that's what we wanted to do here. So the models that I'm going to show you are linear models. Before I plop the charts on the screen, what we did is we have in each of them a sleep or wake variable as the dependent variable. And we're interested in whether PACAP and CORT independently are related to sleep and wake endpoints, and whether their interaction, right, is related to sleep and wake endpoints. We're interested in both the average across those 14 days of measurement and the night-to-night variability. And just to foreshadow, what we're seeing in some of these models is really an interaction of PACAP and CORT. So jointly, when you consider them jointly, they predict some of these sleep bout variables. And I'm showing you here the interaction of the continuous terms, and for display purposes, median splits of PACAP and CORT. And this shows you essentially at the bookends of this chart, the patients who have both low CORT and low PACAP, or who have both low PACAP, high PACAP and high CORT, are the ones who have more sleep bouts, and they're the ones who also have more wake bouts. So their sleep is more chopped up if both are low or both are high. For variability night to night, we see the exact same pattern. The patients who have both low peptides or both peptides are high, they have more inconsistency and more variability of their number of sleep bouts night to night. For duration of the bouts, we don't have as much many findings. There's one interaction predicting the duration of sleep bouts, average sleep bouts, and here high scores on the y-axis is good. It means you're sleeping longer, and again, you see the people on the bookends here who you have both low or both high of the peptides, and your sleep bouts are shorter. So worse sleep. More fragmented sleep when both are high or both are low is what we're seeing. I could speculate beyond my data because it's fun to do. We could suspect that the people who have high values of both are the ones with that enhanced stress state, the revved up HPA axis, whereas the ones with the low values of both, maybe they're the ones that are blunted. Of course, we can't actually say that because we only have one blood measure, right? But that could be a next study. So what's cool about this, I think, if it worked out, is that actigraphy, movement-based, is relatively simple data to collect and scalable, and so is blood measures of CORT and PACAP. So if you could predict which groups patients are in, and maybe it has implications for which medication helps their sleep, that would be pretty cool. And of course, it has translational value. We could also send this back to the animal models where they could also do movement-based sleep and peptide measurements in the same way, and really, truly experimentally test, right, some of these ideas. So thank you to everybody who's here and others who are listed here and funding. And I'll next pass it along to Christine.
Hello. My name is Christine. I am a new assistant professor at Washington State University, just finished my first semester. And I will be talking with you about associations between stress and sleep state misperception among trauma-exposed women. So I will skim over a lot of this because Isabelle gave a really lovely talk covering a lot of the background, but essentially we know that sleep disturbances are highly prevalent after trauma exposure, and it's been posited that hyperarousal, such as heightened physiological or psychological arousal that occurs after experiencing a trauma, may contribute to this difficulty with sleeping. And so in general, we know that stress disrupts sleep, and sleep reactivity is a construct that describes a trait-like degree to which stress exposure disrupts sleep, resulting in difficulties falling or staying asleep. So despite this, evidence of objectively shown sleep disturbances has been kind of mixed. I'm going to conflict a little bit with what Isabelle just said. But one meta-analytic study by Kobayashi and colleagues examined studies utilizing polysomnography in PTSD patients. They did find that patients had more stage 1 sleep, less slow-wave sleep, and greater REM density, although the effect sizes were small. Conversely, Lewis and colleagues conducted a meta-analysis of actigraphy-based studies, and they didn't find significant differences between PTSD and non-PTSD groups on variables such as total sleep time, wake minutes after sleep onset latency, or sleep efficiency. So, like with any other construct, sleep is multifaceted, as we know. So sleep duration is obviously the thing that's discussed most, but we know that there are other components of sleep health that are important, such as how long does it take you to fall asleep, how many times do you wake up in the middle of the night, what percentage do you spend in different stages of sleep. And so accordingly, there are multiple ways of measuring these different facets of sleep. We know that polysomnography is the gold standard, but very taxing on participants and staff. So more ecologically valid methods can look like actigraphy. People in the sleep field have also been utilizing things like Apple Watches or Fitbits, as well as nearable devices, which are devices that are placed near you as you sleep, nothing that you have to wear. And of course, you can do self-report measures to get a subjective report of how people are sleeping each night. Sleep state misperception, or paradoxical insomnia, is a phenomenon that occurs when individuals report their sleep to be different than objective methods assess. So, for example, just thinking that you are awake when objective measures might say that you are asleep. And clinical samples may report a greater degree of sleep state misperception, which could be due to greater distress or bias from experiencing clinical symptoms, but overall contributors of sleep state misperception haven't been thoroughly investigated. So the purpose of this study was to look at how stress-related symptoms was related to the degree of potential sleep state misperception among trauma-exposed women. So in this study, 55 cisgendered women completed the whole study procedures. Here are the broad inclusion and exclusion criteria. For exclusion criteria, we just included things that was related to safety concerns or factors that we knew were significantly likely to disrupt typical sleep patterns. Here is the overall study design. So first they completed a baseline assessment and filled out self-report measures. Then they completed 7 days of continuous actigraphy and sleep diary. And on 2 non-consecutive days, participants collected saliva to assess for cortisol. For the baseline assessment, they completed a brief psychodiagnostic assessment and filled out the following self-report measures to assess for trauma exposure and current mental health symptoms. Then they wore wrist actigraphy for 7 days. I will pretty much skip over this because a lot of it has been discussed already, but here's just like a general overview of actigraphy. Here is an example of an actogram that's shown from the Philips Respironics ActiWatch, and you can use these validated sleep scoring algorithms in order to estimate sleep-wake patterns from activity levels. So using the actigraphy, we generated the following variables: total sleep time, sleep onset latency, wake after sleep onset, and sleep efficiency. For those who don't know what sleep efficiency is, it's basically like how much time you're spending in bed actually asleep. We also use the consensus sleep diary and as a metric of subjective sleep, and we added items assessing for pre-sleep arousal. So in order to do this, we added the pre-sleep arousal scale, which generates subscales of cognitive arousal and physiological arousal, as well as the fear of sleep inventory. So we, and by we I mean I, because this was my dissertation, I modified the questionnaire to assess for how they were experiencing these symptoms on a night-to-night basis, so that we could kind of try to figure out, like, on this night before you went to sleep, how much arousal were you feeling? And we collected 2 salivary samples for 2 non-consecutive days, and this was to assess for the cortisol awakening response. So the first sample was collected immediately upon awakening, the second was collected 30 minutes after, and so that we could try to observe that naturalistic spike in cortisol that you get when you wake up in the morning. So in general, we didn't really observe much in terms of hyperarousal and sleep variables, with the exception of sleep onset latency and subjective pre-sleep arousal. So we found that fear of sleep was positively correlated with self-reported and actigraphy-based sleep onset latency, and we also found that actigraphy estimated sleep onset latency was positively correlated with pre-sleep somatic or physiological arousal. On the other hand, subjective sleep onset latency was positively correlated with pre-sleep cognitive arousal. So in general, having greater arousal made it more difficult for people to fall asleep at nighttime, and this was observed both subjectively and objectively. Makes sense. And while most self-reported and actigraphy-based variables were moderately correlated with each other, we did observe significant discordance between the two methods. So participants tended to overestimate their sleep duration compared to what the actigraph said. And they also tended to overestimate their sleep onset latency. So they said that it took them longer to fall asleep than what the watch said. On the other hand, participants underestimated their wake after sleep onset compared to the actigraph. So the actigraph said that they were awake for a longer period of time in the middle of the night after they fell asleep compared to what the participants said. And in terms of stress-based correlates of sleep state misperception, we found that PTSD criterion E symptoms, which is the hyperarousal symptoms, was predictive of the degree of misperception for total sleep time. And PTSD criterion D, which is negative alterations in cognition and mood, and criterion E symptoms, hyperarousal, was predictive of the degree of misperception for sleep onset latency. So despite the moderate correlations that we saw between the actigraph and sleep diary, we still saw differences between the two methods. We also found that types of hyperarousal were differentially related between the two methods, such that physiological hyperarousal was linked to actigraphy estimated sleep onset latency, while cognitive arousal was linked to subjective sleep onset latency. And so this kind of suggests that the different methods might be susceptible to certain biases or potentially picking up on different things. We also found that PTSD symptom clusters were differentially predictive of sleep state misperception. Specifically, hyperarousal contributed to both overestimating sleep onset latency subjectively and, quote, accurate report of total sleep time. And I say accurate only in the sense that there was less discordance for that. While negative alterations in cognition and mood also contributed to the overestimation of sleep onset latency. And so this raises the question of kind of how accurate the actigraphy estimations are. Conversely, how accurate are people's perceptions of their sleep and what they report on sleep diary. So, is it that clinically significant samples are biasing their reports of sleep because of their distress, or is it that something like an actigraph might not be sensitive enough to pick up on true wake and sleep in this sample? So this study raises some implications for methodology. So first, the findings highlight the potential advantages of a multimodal sleep assessment to capture different components of nocturnal activity. If you are going to opt into picking just one method for assessing sleep, considering the purpose or goals of assessing sleep in your study in the first place. So, for example, are you interested in knowing what the participants' true sleep-wake patterns are? Or are you interested in knowing what types of sleep complaints that people have? If the latter, then maybe just like a sleep diary would be sufficient. And this also points to recognizing the limitations of each method. For example, that actigraphy is simply an estimation of sleep based on movement and not a guaranteed assessment of sleep-wake that you can get from polysomnography. And finally, I think it would be interesting to probe for the potential of machine learning algorithms for the optimization of sleep from wearable devices, particularly for clinical populations. So it might be interesting to compare a sleep scoring algorithm for actigraphy with polysomnography in a clinical sample in order to improve the accuracy of the algorithm for this population. All right, that's it for me. Thank you.
Hi everyone, I'm Khairunisa Ibrahim from the lab of Jose Moran Concepcion, at Washington University in St. Louis, and today I'm just going to share our project of understanding impact of chronic pain on sleep quality using a multi-sensor approach in an animal model, so kind of switch out from everybody else. Just a brief overview. Within the US, there is 20% of the population that suffers from chronic pain, and in this population, there are more than 50% of them that report sleep problems. That includes difficulty in falling asleep, insufficient sleep, as well as sleep quality. In addition, there's also about 20% of the population report at least one symptoms of insomnia. And this is because there's like this pain and sleep cycle when the first onset or presence of pain is uncomfortable, so you have difficulty in falling asleep and also staying asleep. And because you have inadequate rest, you also delay your healing, and the next day you feel worse and you have more pain. When you have more pain, you are more uncomfortable, and again, the cycle continues. You have increased difficulty of sleeping and staying asleep. So this whole cycle keeps on continuing, worsening your pain, and also worsening your sleep quality. For this project specifically, we are interested on the first stage is when the onset of inflammatory pain starts to impact the sleep. So in an animal model, we induce inflammatory pain by injecting complete fluid adjuvant or CFA into their hindpaw. It is an inactivated mycobacteria in mineral oil, and it does induce inflammation. So as you can see here, animals that inject with a vehicle, which uses saline, their paw is not as inflamed as animals that inject with CFA, and this thickness in paw is significantly different from control paw for up to 6 weeks. It also induces long-lasting hypersensitivity, so whether you're looking at mechanical hypersensitivity or thermal hypersensitivity, it will, paws that have been injected with CFA shows a decrease in paw withdrawal threshold or latency respectively, suggesting that they are feeling more pain. As I mentioned, this project includes a lot of approaches. So the first approach that we did was to use a passive infrared sensor. This is just to quantify the circadian rhythm of the animal by detecting the locomotor activity of the animal. It is high throughput because you can connect up to 400 cages to one computer at a time. So it's simple and it's low cost to start with. This is an example of the readout of the locomotor activity at each individual time point. And at this stage, I also want to highlight that the mice are nocturnal, so their active period is when lights are off, so you can actually see high locomotor, and when lights are on, they are actually technically supposed to be sleeping, so you see low locomotor activity. The next approach was to use the piezoelectric sleep monitoring system. This is to quantify the sleep quality by looking at the sleep-wake patterns. It is non-invasive. It's also an automated system. It detects the breathing rate frequency of the mouse in their home cage. It is so sensitive that it can differentiate when the mouse is actually awake versus when it's asleep. So this is just an example from their website where from the breathing rate frequency, they will score this as the animal is awake, and they will score the bottom two as the animal is sleeping. And in a clinical setting, just to bring it back, it is similar to all your smartwatches that you wear in order to detect sleep-wake patterns. And the last approach that we did was the wireless EEG and EMG electrode. This is to quantify the different sleep states by recording and using your delta or theta power recorded from the brain, the muscle tone, as well as the animal activity. It is definitely more invasive because you have to implant the electrode into the animal. And this is an example of the parameters that is recorded from the electrode for them to quantify the different sleep states. And again, bringing back to the clinical setting, this is like the EEG head caps that the patient would wear in the clinical studies. So how do we use all these different approaches in the current project? So the first one was to use the passive infrared sensor. We wanted to study the impact of CFA on the circadian rhythm. So it's just a very simple device clipped onto the top of the metal grid of the home cage. And they have their own software called Clock Lab that allows us to record parameters such as percentage variance, relative amplitude, interdaily variability period, FFT amplitude of the circadian rhythm itself. So for the sake of time, I'm just going to focus on two, which is relative amplitude and interdaily variability. So relative amplitude is just to assess the 24-hour rest activity rhythm of the circadian rhythm. And the interdaily variability is to assess how fragmented your circadian rhythm is, or actually the animal circadian rhythm is. Animals are put in regular 12 hours light-dark cycle. We record baseline for a week before we inject saline into the left hind paw, follow up for a week, and then we inject CFA into the right hind paw. Interestingly, what we saw was there was no CFA effect on relative amplitude. Maybe a little bit of CFA effect on interdaily variability, but that's not significantly different from when they were injected with saline, and more likely significant from their own baseline. So at this juncture, we were wondering if the circadian rhythm has been entrained by the environment lights, such that it's masking the effect that we might have seen with CFA. So we repeated the experiment where the animal is in constant darkness. So we remove the environmental light that might have entrained the circadian rhythm. And again, interestingly, we did not see any CFA specific effect on the relative amplitude or interdaily variability, or rather any of the other parameters that we were recording and analyzing, so suggesting that the CFA had no impact on circadian rhythm. So the next thing we wanted to know is whether CFA had an impact on the quality of sleep. So maybe the animal is low in activity and it's being, circadian rhythm is still intact, but when it's low in activity and technically supposed to be sleeping, is it getting quality sleep? So we move into the piezoelectric sensor. So like I mentioned, it's a very sensitive platform. You just put the home cage on the platform and it will be able to detect the breathing rate frequency of the animal and differentiate when the animal is awake versus when the animal is sleeping. Animal is still in their regular 12 light-dark cycle, and we record baseline before we inject the saline and CFA respectively at their time points. And when we were looking at the recording in the light phase, when the animal is technically supposed to be sleeping, we saw that there is an increase in sleep duration, although it's not very specific to CFA injection. But when we look at the sleep bulk lengths, especially when the animals are experiencing CFA for the first 3 days, we see that there's a decrease in sleep bulk lengths. Although all of them are sleeping more, but the sleep is more fragmented, so they're just like napping. In the dark phase, where they are supposed to be active, animals are experiencing CFA in the first 3 days are actually sleeping way more, although there's no CFA-specific effect for the sleep bulk length. So this is telling us that CFA does induce an increase in sleep duration as well as a more fragmented sleep. So next, we wanted to know what sleep state is impacted by the injection of CFA, and that brings us into our last approach, which is to use the EEG-EMG electrode. And after implantation, we allow the animal to recover before going through the experimental timeline. Something I want to highlight here for this experiment is that the injections are at the start of lights on. So that means the first 12 hours after CFA injection is during their lights on period, and the next 12 to 24 hours is during their dark period. And the parameters that we can analyze is REM duration and bouts, slow wave sleep duration and bouts, and wake duration and bouts. And again, I'm just going to focus on REM duration and slow wave sleep duration. So within the first 12 hours after CFA injection, in the light phase, we saw no effect on either REM or slow wave sleep duration. But interestingly, in the next 12 hours in their dark phase, we saw a significant increase in both REM and slow wave sleep duration. So again, at this juncture, we took a pause and we were wondering, does it actually take 12 hours for CFA to come in effect and affect sleep, or the effect of CFA is only predominant in dark phase? So to answer that question, we actually repeated the experiment, but this time around, we injected the animal at the start of lights off. So it means that the first 12 hours is now in their dark phase, and the next 12 hours is in their light phase. And we saw that in the first 12 hours in the dark phase, we already see that increase in both REM and slow wave sleep duration when the animals are injected with CFA. And in the light phase, we only saw it in the slow wave sleep duration, but no longer in the REM sleep duration. So putting this all together, it seems that CFA does increase both REM and slow wave sleep duration, predominantly in the dark phase, regardless of when the injection happens. So just to summarize all the approaches that we have and what is our conclusion, we first started with a passive infrared sensor to study the impact of CFA on circadian rhythm, and we saw there was no impact. We then moved to the piezoelectric sleep monitoring system, and we found that CFA does increase sleep duration, and it also leads to them having more fragmented sleep. And then we move into the wireless EEG and EMG electrode to understand which sleep state is actually affected when we see an increase in sleep duration. And we saw that it is both the REM and slow wave sleep duration that is increased, and this is predominantly in the dark phase, regardless of when the CFA is injected. And altogether, these approaches that we have in a preclinical experiment helps us to validate our current animal pain model so that we can move on to future sleep studies where we investigate the mechanism that might underlie sleep disruptions in the presence of chronic pain. And of course, the very, very big picture why we do this in preclinical settings and trying to find the mechanism underlying all of this is so that we can provide mechanistic insights in developing future therapeutic strategies to address sleep disturbance in chronic pain patients, but really very, very big picture. And as I mentioned, there is a ton of parameters that were analyzed that I can't really go in depth in this presentation, so I will be at a poster session later this evening, and I'll be happy to chat more with any of you on any other parameters that I have not discussed today, so please do stop by. And lastly, I'd like to acknowledge the people that put in hard work in this project. Dominica Brick, who's from the lab of Bill Carlezon. She is the main driver of the EEG-EMG experiment. And then I have Justin and Ashish who work with me in Washington University on the circadian rhythm as well as piezoelectric experiment. Ashish is from the lab of Eric Music, so in Washington University, we also have a sleep core, and Eric Music runs that sleep core. I'd like to also acknowledge the support I get from the JMC lab, as well as our funding agency, because without them, we will not be able to do this work. All right, thank you.
Hello, everyone. I'm Rahimi, instructor at McLean, and I'm going to talk about the sleep parameters that we can get out of the actigraphy data, and some of the correlations or some of the inspirations that we can get especially for the depression and anhedonia in different data sets, and including the trauma-exposed data. So Isabelle, Christine, and Khairunisa made my work much easier. I don't need to convince you that sleep is important, especially in Saturday afternoon. So we want to get the parameters of sleep. Parameters of sleep includes the sleep patterns, sleep quality, and even activity. Activity during the day is not very different, and there are a lot of literature about this, that these are very important. Having a good active day will associate with a good night's of sleep. So what we are trying to do is using the devices, including the actigraphy data and phone. Here I will be talking about the actigraphy data, but that they are giving us some signals. The actigraphy data only gives you as a raw data only the accelerometer data, as you see. And then what we want is is the sleep pattern. You are seeing also GPS there. I'll tell you why. So for the actigraphy data, we have developed this algorithm. From the beginning, when we started doing this research, we decided to work with the raw data because you will always have a good control that what you are inferring and what are the features that you are extracting. There's always some models behind that at the end the analysis will be affected by that. So it's important to have a good control over the data, and that's why we use the raw an actigraphy device that gives us the raw data. One of them is the gene active, and then developed a pipeline. We call it DP sleep, deep phenotyping of sleep, and the one important thing Isabelle alluded to, that polysomnography is defined as the ground truth of sleep. But in these deep phenotyping projects, we are tracking participants for years, 2 years sometimes. So you see the problem. How we can track that? And this is probably, we can call it that this is a more natural way of the way that people are sleeping. So this deep phenotyping pipeline gets the raw data, and and in the first step we are getting something like this, that is the pattern of activity. You are seeing blue as low activity, red as high activity, and that shift there is actually the time zone change. So that's why we sometimes, for some of the projects, we also need GPS data to correct that time zone. You see here, this is the sleep that you are detecting without correcting for time zone, and this is the sleep when you are correcting for time zone easily with just the GPS data when you are combining those. Fortunately for this project, which for the PTSD project, spared project as it's called, because it was 2 weeks, we didn't need the GPS. But when it goes to 1 year, especially for students, this is a student, they go for break, and you see that immediately there. But for some projects, you need it. So one of the places that we applied this was the UK Biobank. There are a lot of papers and projects on UK Biobank. They applied that there. And for UK Biobank, we are fortunate that they came back in 2015. They collected 100,000 samples of 1 week of actigraphy data that we are able to analyze those. These pages here that you see is these actigraphy that I personally went through those hundreds of those pages. And this is very similar to, when you look at it now, it's very similar to looking at the brain images. I was getting kind of expert to do that, and my speed was increasing when I was doing quality control. So this is one of the messages of this talk, that quality control in this data is the key. And that's why having tools for that, so that you can do quality control in big scale, is very important. So I'll go a little bit faster about what we got from the UK Biobank data, but it is also important to see. For example, so what you are seeing here, the UK Biobank data is actually for age. So 44 years up, and you are seeing the. Let me use this. So you are seeing the age, the 5 being age, and immediately what you see is the activity drops from the more red here goes to more green, and the activity during the day of the participant, based on the year, and these are 70,000 participants out of that 100,000 that we QC'd. And the other pattern that we saw, that you will see that later, is something that is called social jet lag. Social jet lag is an important concept in sleep, that usually during these are the days of the week, Monday to Sunday, and Monday to Friday, usually people are sleeping less, sleep duration. It's different between men and women, between male and female. But the interesting thing is, in most of these patterns, especially before the retirement, you are seeing the change between weekday, weekend, and after that, when for older people, for older individuals, we are seeing that it goes to a kind of an equilibrium, that the weekday, weekend is not important, is not very different, and it has its own lessons, that how men and women are based on that baseline. If we define that as the equilibrium, they are sleeping, actually, and how they are kind of sleep deprived, probably. But this pattern, we expect to see that in most of the patients, in most of the individuals, and we are actually experiencing that. The other thing that we were inspired by in the UK Biobank is they also asked about their depression symptoms, and they were reporting the ones that they are reporting depression. We are seeing that their wake activity is consistently lower. So this is another one that you will see that the activity is dropping, but we are seeing that it is lower. Based on these, the parameters that we can get from the sleep, from the actigraphy, including the sleep duration, the quality of sleep, the wake duration, and the activity during the day. These four can drop into one model that is, we call it a battery model, so that the patient, we are kind of getting more energy during the sleep, and based on that, and we are using that energy during the day. And if we consider that as an accumulative model daily, this can define that. I'll go quickly through this patient. That is, I told you that actigraphy data can be collected for 2 years. This is 700 days of one participant, N of 1. And this patient is having a clear fluctuation of mood, depressive episodes of about 100 days. And during these 100 days of depression, we are seeing more sleep time, bad sleep quality, and the lower activity. Here you see the orange. The orange is lower activity during the day. So that's why these two can come together, and we can define that. But what we see in the trauma-exposed patients, these are all just the parameters that can inspire us, that can inspire us what to look into. So these are the trauma-exposed patients. These are activity data, and you see that these are not perfect. That's why we need quality control. Here you see that they are not wearing the watch. Some of them are only wearing it for 1 day, and some of them even didn't wear it. When we do quality control for the perfect ones that they were supposed to wear it for 2 weeks, you see most of these are 2 weeks. I just passed some of those that they are giving us 1 week, because the weekly pattern was important to us. So the lowest requirement was 1 week of wearing the data, wearing the watch. And here you can see I just go back and forth. Just focus on the blue and the activity, and how we can do the quality control, how we can see even some patients that probably they are night shift worker, and if we want to include or exclude those data. So I think that's again exactly the message that how we can play with this data. The rest of these, 129, Isabelle showed more, because for those analyses, even 3-4 days of data was enough. I included the ones that have 1 week of data. And here is the pattern. I just divided the patients because my interest was looking into the mood and anhedonia. I just divided the patients based on the SHAPS score, SHAPS 14 score, to see if there is any difference. But the only pattern that was first important to me was especially looking into the wake time, sleep onset and wake time, if we are seeing that pattern, if we are seeing the pattern of social jet lag. It should be there, except for the inpatients. We didn't see that there. But when I'm adding more data from different studies, here you are seeing the schizophrenia healthy and clinically at risk, cyan and magenta. Also, the bipolar patients in green, you are seeing that their sleep duration is also higher. But all of these are following the pattern. And one other group that is important is the college students, the data from college students in black. That is, their sleep duration is lower, but also, and I'm glad that the sleep fragmentation is confirming. Sleep fragmentation for the college students, you see, this is the lowest. And for each of these, when we are comparing those, not significantly, but you can see the difference. And also, we are seeing that these patient population have more sleep fragmentation, which was confirming. I also did the stats analysis controlled for age for the SHAPS and fragmentation, and especially in the female population, it was more significant. So, to summarize, actigraphy data can give us some objective insights that we can use it, especially for the sleep patterns, sleep quality, and the daily activity. Age and sex are important to consider, and also the sleep parameters can tend to have a weekly pattern that needs to be considered in different analysis. And also, what we are looking into is this poor sleep quality, which is higher sleep fragmentation, later sleep midpoint, that Christine was talking about, that sleep latency, that we'll talk about that in the discussion, can be also important, and the reduced activity during the day are important. Thank you very much. I just want to thank for Isabelle for letting me work on this data. Justin Baker and Randy Bachner, I developed these pipelines in their labs, and it was a joy. And thanks for Sydney and Aylul and others for quality control most of this data.
Okay, first of all, I'd like to thank Isabelle for giving me the opportunity to contribute to this panel, and to congratulate each of the speakers for really wonderful work and very clear and concise talk. So thank you. On behalf of the audience, thank you. Before taking questions, I just wanted to emphasize how important it is to better align procedures and endpoints across species. I personally believe that this is the path to more effective use of animal models to predict outcomes in humans, which is one of the main reasons why we use these animal models. A lot of the methods that we use to study psychiatric illnesses in animal models were built for speed and efficiency in the drug discovery process. So drug companies using these tests or assays to make rapid and inexpensive decisions on which drugs to invest in and to continue studying. And as neuroscientists, we've adopted a lot of these models into our labs. But I think we're learning from experience that that approach is not optimal. The pipeline for mechanistically new drugs that have been developed over the years for psychiatric illness is very, very sparse, so to speak. So I think it's a time for new approaches, and I think aligning the endpoints that we use in our animal models and humans is something that we really have to focus on. So we've written about this in a recent review paper that we published on PTSD. I recognize that that's small, that little box there, but I provided the reference if you wanted to go to that paper and look what's written there in detail. But I'll summarize the main points here. We want to use similar or identical endpoints across species. So if you can't measure struggling behavior in humans while they're suspended upside down, you probably shouldn't be using that as a model. The tail suspension test is what I'm referring to in mice. Sleep is a good example of a translationally relevant endpoint because it's defined and measured in the same way across species, even though obviously humans and mice, for example, sleep in different patterns throughout the day. The bottom line is that what is happening is that sleep is measured and defined in exactly the same way, so you can compare across species. It's also important to be able to use continuous data collection. So a lot of procedures in animal models depend on measurements of a single and often arbitrary endpoint because it's defined and measured, I'm trying to read from my tiny little notes here. Sleep is an example of a translationally relevant endpoint because it's defined and measured in the same way across species. Using continuous data collection, a lot of procedures in animal models depend on measurements at a single arbitrary time point. I use the analogy of a cardiologist going to a cardiologist and the cardiologist making the diagnosis on the basis of a single heartbeat. Obviously, that's not what happens, as opposed to looking at an EKG. So to be able to make these measurements over long periods of time is really powerful, and I think Habib, you showed a beautiful example of that. Who wouldn't love to have 200 days of data from a mouse or 2 years of data from a human to be able to detect these patterns and the effects that our manipulations are not having on a very arbitrary endpoint, but in patterns that occur over time. It's also important to be able to use objective measurements, exactly the type of approaches that Sam talked about in his keynote. So no more scientists with white coats and a clipboard making little check marks that are based on really subjective impressions or judgments. We want to be making really objective measurements that are reproducible, not only across members of a lab, for example, but even the same lab member going back and seeing exactly the same thing each time. And finally, using endpoints that are increasingly becoming available by advances in the development of smart devices and wearables. So I think that these principles should hasten the process of turning discoveries that we make in the lab into new treatments, which is something obviously we're all here because this is sorely needed. So with that, I'd like to take questions if people have questions in the audience, and I feel like I can almost call on people by name. Kate?
Thanks. This was a great panel. I have a question, I think, for Isabelle and for Christine, two very separate ones, but maybe I'll start with Isabelle. You mentioned the difference between the high groups and the low groups, and I was wondering if you have data that can kind of separate those groups out, whether it be kind of PTSD, years of diagnosis or previous the life events checklist, or even current daily stressors that they might be experiencing that could help explain those two groups?
Yeah, I mean, we have a lot of, self-report data on their history and other self-report data. I haven't looked at that yet. I know that they're not different in terms of their severity of PTSD symptoms and their depression. And that, if we use the self-report measures of sleep in these models, that we don't see anything. But I haven't yet, taken these data this way with, okay, let's group these patients this way and see if they differ on other variables like, one of the ones that would be interesting would be history of trauma cumulatively. I think actually in some of Carrie's you're back now. Carrie's publications, in heavily traumatized PTSD patients from certain backgrounds, you see certain relationships with stress and we don't see all of them in this sample. But anyway, I think looking at that, comparing their their early life history and whatever we have, because it's not as rich as what some people are doing now with neighborhood disadvantage but we have some data on history of trauma that I think would be interesting to look at, whether really we have, some folks who have really that chronic early trauma, and whether that's what we're seeing. That would be nice if that worked out, but I haven't looked yet. I don't know if that answers your question.
Yeah, that does. I will look for, I'll follow up on that and see what you discover. And then Christina, I guess I'm curious too about the, when you talked about like actigraphy being an estimate, right? I think that is kind of important to keep in mind when we think about treatment outcomes and seeing how treatments are moving the needle on sleep. And at a certain point, even if you have really good actigraphy data suggesting that sleep is improving, if the patient's perception of sleep isn't improving, could you talk maybe about is there any data to suggest that giving participants that feedback about their sleep, or patients their feedback about sleep, improves the perceptions? How can you move the needle on that, the discrepancy between it?
That's a really good question. I am not sure if there is any data on that specifically in terms of giving the feedback. Clinically, I'm a sleep psychologist, so clinically, a lot of people want to bring in objective data and say, look at what my watch is saying, and those kinds of things. And then, but when people are coming into the clinic, kind of the subjective information is what's important, because that's what you need to make a diagnosis for sleep-related problems. But I think that it kind of ties back into potentially the underlying reasons as to why they think that their sleep might be disrupted. So, if their watch, let's say that we really optimize the sleep-wake scoring algorithm so that we know it's super duper correct, and yet they're still complaining that they're fatigued or, not sleeping well or whatever else, kind of trying to do trying to maybe obviously provide psychoeducation, but trying to figure out the underlying causes of maybe separating fatigue versus sleepiness in general, and then trying to figure out what might be contributing factors to the fatigue. Is it the ongoing mental health concerns, or is it some underlying sleep-related thing that hasn't been adequately addressed, or is it that they're anxious all day and they're exhausted? So kind of trying to do a deeper understanding for what might be contributing to those subjective complaints.
That's great. I wonder if I could do a follow-up to that. To me, sometimes some of the most interesting things in our field are when the objective and subjective don't align, right? And we see this all over psychiatry. You see it in pain. You see it in, I was just at a thing for Joe would do, and it's sort of fear versus threat. But most of those things, it's hard to get a ground truth of what the real pain experience or the real threat experience is. Whereas here, we really have something, we have this very concretely defined sleep. And I wonder if you got any of you human folks or others are very explicitly using that delta between the objective and the subjective as a variable in and of itself. And is that predicting resilience? Is it predicting other things? Is it, as you say, can you use that to align in population studies with some of these other components of psychiatric distress that may be helpful variables in and of themselves?
At least for me. Sorry, I just started. We're all awake now, right? At least in terms of me, this was the first time that I kind of delved into looking at this discrepancy, because we did have the nice one-to-one comparison of sleep diary and actigraphy. There have been other researchers in the sleep and circadian field that have been looking at the difference between subjective report and objective estimates. But you're right in that. I feel like it provides us some sort of clinically meaningful information. And even when we're looking at something like PSG that's considered to be a gold standard, we still see this discrepancy in PSG studies as well. So why are people saying that they're awake when PSG says that they're asleep? Is there something that is actually going on that's contributing to these symptoms or this feeling that might be better understood when we look at like microarchitecture and not just sleep staging percentages. So I feel like it's a very new area that I'm hoping that people will be motivated to tap more into. But yeah, I think that there's something that's worth probing and trying to figure out what might be going on.
Yeah, sorry about that. I think you've been touching around it, but just to be explicit, like you're looking also at type and timing and chronicity of the PTSD episode or episodes in terms of, because I never heard of paradoxical insomnia. Like what would lead to that?
Can you repeat your question? I'm so sorry.
Or I guess it's for both you and Isabelle, but kind of subdividing these sleep phenotypes into type and also onset of PTSD, because I'm in the early life stress field.
In my study, I can only speak for that. In my study, they had to have a requirement of being trauma exposed in childhood, specifically childhood sexual abuse. That was like the larger study that we were looking at, and this is just a subset. So in my sample, they didn't have to have a diagnosis of PTSD, but they did have to have childhood trauma exposure before adolescence. So that was consistent across my sample, and unfortunately, I didn't have like a control group to in order to do a comparison.
Yeah, so I didn't take the time to talk about this, but our sample is also not all full PTSD diagnosis, so we have a kind of a range of PTSD symptoms across the board. I think we could look at, and I feel most of my studies, at least, people have had chronic PTSD. I haven't seen a lot of people who I don't know if it's the way we're recruiting them, but we could, look at duration of illness and try to document remitting and relapsing course, but we haven't really done that. We generally interview them to see if they meet current criteria and what their current criteria, and then get a sense of when do you think this started or how long after the trauma did it start. So, I think we have some variables that get to chronicity of illness, but I don't know about subgrouping them into chronic versus, I don't think we could do that with our data. And the other interesting, loose association that I'm having to this is that this becomes a stressor, right, itself, if you're chronically ill. So when we're studying stress, we can think about it that way too, in terms of chronicity of stress exposure.
Sam has a question?
So I guess this is a more maybe conceptual question for the whole panel. So when we're using mouse or rodent models as our proxy here for the human condition, and we're using nocturnal versus truly like more human-centric diurnal model organisms, I'm just wondering if there has been any, or how you all think about if there are better model organisms other than mice that we might be using, still within the rodent sphere, for capturing a more translational state.
That's a very good question. I feel like if you're still within the rodent model, I'm not sure how we can move past that nocturnal.
Squirrels.
Thank you. Yeah. Yeah, I guess that's will be one way.
Well, I guess maybe as another way of wording it, for preclinical researchers, what might be the major bottlenecks or concerns we should be thinking about in a nocturnal model if we're trying to maintain translationability of the work?
Well, I guess the behavior, we will have to take it with a pinch of salt, I guess, because because the way they sleep and their patterns are different, whatever we see with, I guess if I bring it back to my experiment with like chronic pain, even their experience and perceptions of chronic pain and how we measure chronic pain in these animals is also different with how we would measure it in human studies. And that putting together with sleep and the way their sleeping patterns and their circadian rhythm cycles is already different would give us some insights, but probably not the full story, which is why I guess we are doing all the different sensors, because when we first started, we were just looking at locomotor activity. Like when I first started this sleep study, we were doing deep black gut. We were just recording the mice in deep black gut and just looking at, okay, is the mice moving, is the mice not moving? And then we decided that it's not capturing the full story of sleep, because in humans, you don't just look at whether they are moving versus they are not moving. And that's when we move into those multi-senses approach to get a sense. So it's just the start of it. But again, I acknowledge that is like that difficulty in trying to fully translate to humans, because we're also looking at chronic pain, and chronic pain itself, we also have that question, like how do we actually translate how we test that animal is feeling the negative effect with chronic pain or the nociception with chronic pain, and how we translate it to the human as well. So it's a good start to get mechanistic insight, but it's definitely a good question to think about on how we actually fully translate it to human.
Good luck with your squirrel research program with the new administration.
I'm thinking about it hard.
Before you brought up squirrels, I think that's a really cool idea, actually. I've always wondered how much of the way that humans sleep is really dependent upon the way our brain works, as opposed to kind of when the lights are on and when we can sleep and when it's dark. Are we kind of culturally entrained to this pattern of sleeping? I don't know the answer to this, but.
Yeah, I was specifically thinking of like the fear and food entrainment literature in our mice models, right? Because you can get really nice fear entrainment in mice at different parts of either the onset or versus offset of the light cycle. And I do know that that's been hard to replicate. Or my understanding is that in the literature, that's been hard to replicate in the literature. So that's like sort of the question I have. When I think about sleep fragmentation in C57s, like inbred C57s, versus sleep fragmentation in the human literature, or even sleep fragmentation in outbred mice that have more genetic variability, I'm not sure they align well. And so, if we're using sleep fragmentation as our metric, as an example, should outbred mice versus another, versus inbred mice, or versus another model organism altogether be more appropriate to mimic what we're more naturally seeing in humans? I don't know, C57s are weird mice in general. Like there's a whole nother soapbox to get up on there if I wanted to, but especially for sleep, it does beg the question, where should we be careful when making these comparisons with an inbred C57 versus human or other outbred lines? And I don't have an answer. Like, I don't know the answer, to be clear.
Thanks.
Other than squirrels, yes.
Sabina Beretta?
I was just going to say it's not my work, but what you brought up is actually really interesting. There is evidence that humans not always slept through the night, right? That they were taking a nap, they woke up, they did stuff, they visited neighbors, slept a little more. So it could be cultural, which could be even more difficult to mimic or parallel in an animal model.
Right.
I have a quick question. So I thought it was interesting for Dr. So's work that it was in females, which I'm now understanding was part of the inclusion criteria, but it sort of got me thinking, this is a question for anyone here, about like sex differences in sleep disruption as a result of trauma or pain, whether it's in mice or humans. And I'm just curious, like, have you found any differences between males and females in those responses?
So for the basic the experiments that we do have include both males and females. We try to see if there's a sex differences, but we didn't see. So all the data that we saw was pulled. There are some papers that kind of suggest that the females tend to be more affected by chronic pain, and that's what we were looking for when we were looking at chronic pain and sleep, the impact on sleep quality. But at the current standing of our data, before we like do more in-depth analysis, we don't see that sex differences yet.
Yeah, I can talk about the sleep duration and sleep timing, especially because it's very hard to these cohorts that we have. They are very small to infer those. But for the UK Biobank, what we saw was usually women sleep more than men. So, and there's an interesting discussion about that, and the difference is because usually the wake time, especially when they are working, before retirement, the wake time is enforced by the society. The onset sleep for men is later than women, and there's this interesting discussion that it's not necessarily because of the tasks. It's more about leisure. So they just want to go to bed later and go to sleep later, and this is something that you are seeing that. And after that, when later time when they are older, they reach to the same equilibrium. But before that, it's a kind of probably you can call it a social sleep deprivation.
Yeah, that's really interesting because I don't think we've ever reported this, but we've looked at sex differences in mice, just how they sleep, and female mice sleep 45 minutes more in a 24-hour period. We haven't done anything more with that. In a lot of the responses, we don't see, when using sleep endpoints as a metric, we don't see differences, but the females are sleeping more. While you have the microphone and everybody's eyes are on you, I don't know if you've been properly acknowledged in other settings for all the work you did to put together this conference, so thank you.
Thank you.
Washu, did you ever try all three of these or any two combinations of them in the same animals and at the same time? You know, I guess that's one of the questions. How do the different metrics of mouse sleep compare to each other in terms of you know, actigraphy versus EMG or whatever? Just any thoughts on that? And if you haven't done it, that's fine. But.
So in this experiment, we do it in different animals for all three of them. But for the sleep core, that they have done passive infrared with the phasal sensors, and they found that they do, depending on how they manipulate, but I think for baseline they do correlate. I also know Ashish is also now working on correlating the phasal sensors with the EEG. Of course, the phasal sensors is not accurate because, you know, although they sell it that it is accurate because they're using the breathing rate frequency, but there is definitely some differences. Obviously, the EEG is the gold standard, but it is pretty close. So it has been done, but not in the current project.
Any other questions?
I have a question. Thank all of you guys for these talks and this great discussion afterwards. This is also kind of a bigger picture, more conceptual question that I'd be interested in an input from any of you guys. So all of you used actigraphy in your studies. But wearables are really gaining lots of traction, and there are a million now, right? Fitbit, Aura, Whoop, Apple Watch, where you can also get these sleep metrics. Now, one of the tricky parts of that is that you don't get that raw data, so what happens to derive those different sleep metrics is a bit of a black box. And at the same time, wearables really do provide this amazing opportunity because so many people are already wearing them, and you can give people these devices and they will wear them for months or years, right? So you can really look at intra-individual variability in this really interesting, meaningful way. So my question is, where are we in terms of independent validation of some of these wearable sleep metrics, or what kind of data would you want to see that would make you feel more confident to use these devices in your own work?
Yeah, it's a great question. It takes probably 1 hour to discuss that. Yeah. We are not somewhere that we are at least we say that close to converge. That's any of these devices. We still think that a lot of those devices, when they are extracting the sleep, there are a lot of models behind that we don't know. That's the problem. When you say actigraphy, is this exactly actigraphy or accelerometer? Because we know that Apple Watch does that, but a lot of that is based on heart rate and heart rate variation. The same is for other devices. So it is, and based on the application, it is very important that what we want to do with those data. I think what I want to see is all of these different devices. Empatica is another one, another great one, that also you can get the raw data. They also provide you with the sleep, and they're using different modalities of the data. Empatica, gene active accelerometer. The gene active accelerometer are already giving. All of them actually give the raw data that they are using for their inference, and they are compared with each other. Actually, what is actually used for those, and probably some pipelines, exactly like all of the pipelines that, for example, now are used for the connectivity and brain. Why not using it for these devices? That just give us the raw data that you are using with the same granularity, and then probably we can compare those and trust. Even we don't need probably PSG, just comparing and seeing that they are all converging at the same point. Probably that can help.
Yeah. Just really quickly, there are some people who are using commercially available devices for the evaluation of sleep. Like Michael Grandner at the University of Arizona has fully embraced Fitbits, and he's like using them in his research now. So there has been research validating Fitbits with like PSG, for example. But I think the only downside is that, and I personally am trying to do some work with Apple Watches in sleep. The only downside is figuring out, like, the research grade actigraphs can go for a long period of time without charging. Apple Watches need to be charged every like 12 hours or something like that. And so there's like these limitations to then consider, and then just thinking about what are the just thinking about like what are the pros and cons to each method. Obviously, the research grade devices are easily accessible. People can use them. On the other hand, they provide feedback. So then that's kind of something that to keep in mind. Where, you know, do you want someone to know how they slept the night before and have that kind of bias, how they, you know, what they do the next night and things like that. So it's really just kind of like thinking about what you want to do with the research.
Another anecdote, there's another question back there, but I'll share an anecdote I've heard from clinical colleagues is a lot of people don't like to wear watches at night, and they find them uncomfortable, and they'll take them off. So there may be solutions, there are ring type things, but then you're going to lose data.
We got to wrap soon too. The keynote starts in a couple minutes.
Oh, okay. One final quick question,
Yes.
and we do have to wrap it up, I think, so.
Yeah. So my name is Michele Ferrante. I'm a program officer in IMH. I fund digital technology, digital mental health. We do not fund regulatory science, but I wanted you to be aware of the fact that the FDA does. It's called BAA. So if you have studies regarding like comparing different watches and their performance, or different phones, or different digital technology, that could be done there.
Thank you very much. Okay, I think we can wrap the session. Thank you.